The enigma of cancer. A biological puzzle. Cancer, a word that evokes fear and uncertainty, is not a single disease but rather a collection of over 100 different diseases, each with its own unique characteristics and challenges. These diseases can affect almost any part of the body, from the blood to the bones and from the skin to the organs. Each type of cancer behaves differently, requiring specialized approaches for diagnosis, treatment, and management. What unites these diseases is their origin, the uncontrolled growth and spread of abnormal cells. These cells defy the normal checks and balances that keep cell growth in check, leading to the formation of tumors and the potential to spread to other parts of the body. These cells, once part of a well-orchestrated biological symphony, break free from the normal constraints of growth and regulation, wreaking havoc on the delicate balance within the body. They invade healthy tissues, disrupt normal functions, and can ultimately lead to life-threatening conditions. Understanding the biological underpinnings of cancer is crucial in our quest to unravel its complexities and develop effective treatments. Researchers around the world are tirelessly working to decode the genetic and molecular mechanisms that drive cancer, hoping to find new ways to combat this formidable foe. At the heart of cancer lies a profound disruption of the fundamental processes that govern cell behavior. This disruption can be triggered by a variety of factors, including genetic mutations, environmental influences, and lifestyle choices. Each of these factors can contribute to the initiation and progression of cancer. This disruption arises from changes in the very blueprint of life itself, our genes. Mutations in specific genes can lead to the loss of normal cell control mechanisms, allowing cells to grow uncontrollably. By studying these genetic changes, scientists hope to develop targeted therapies that can specifically address the root causes of cancer, offering new hope for patients worldwide. Genetic mutations, the seeds of uncontrolled growth. These tiny changes in our DNA can have far-reaching effects on our health and well-being. Our genes, composed of DNA, carry the instructions for building and maintaining our bodies. They are the blueprint for every function and structure within us. These instructions dictate how cells grow, divide, and interact with each other. They ensure that our bodies develop correctly and function smoothly. However, these instructions are not immune to errors. Mistakes can occur during DNA replication, leading to mutations. Just as a typo in a recipe can alter the outcome of a dish, mutations in our genes can disrupt the normal function of cells. These disruptions can lead to a variety of health issues. These mutations, often triggered by external factors like radiation or chemical exposure, can also arise spontaneously during cell division. Environmental factors play a significant role in the mutation process. While most mutations are harmless, some can have profound consequences, particularly those affecting genes that control cell growth and division. These harmful mutations can lead to uncontrolled cell growth, a hallmark of cancer and other serious diseases. Oncogenes accelerating the engine of cell division. Oncogenes, derived from the Greek words for tumor and origin, are mutated genes that promote cell growth and division. In their normal, unmutated state, these genes, known as proto-oncogenes, play essential roles in regulating cell growth and development. However, when mutated, these once benign genes transform into cancer-promoting agents. One well-known example is the RAS gene, involved in transmitting signals from outside the cell to the nucleus, where it influences cell growth and survival. Mutations in the RAS gene can lock it in a permanently on state, leading to uncontrolled cell proliferation. Tumor suppressor genes, guardians of genomic integrity. These genes are essential in maintaining the stability of our genetic information, acting as vigilant protectors against the chaos that can arise within our cells. In contrast to oncogenes, which act like accelerators, pushing cells to grow and divide uncontrollably, tumor suppressor genes function as brakes. They keep cell growth in check, ensuring that cells only divide when necessary and preventing the formation of tumors. This balance is crucial for the proper functioning of our bodies. These genes, often described as the guardians of the genome, play critical roles in several vital processes. They are involved in DNA repair, fixing any errors that occur during cell division. They also control the cell cycle, ensuring that cells only proceed to the next stage when they are ready. Additionally, they are key players in programmed cell death, also known as apoptosis, which removes damaged or unnecessary cells. One of the most important tumor suppressor genes is TP53, often referred to as the guardian of the genome. This gene is crucial for preventing cancer as it can detect DNA damage and respond appropriately to maintain cellular integrity. TP53 acts as a cellular emergency brake, halting cell division in response to DNA damage. It initiates repair mechanisms to fix the damage, ensuring that cells do not pass on genetic errors to their progeny. This function is vital for preventing mutations that could lead to cancer. If the damage is irreparable, TP53 can trigger apoptosis, a process that ensures damaged cells are eliminated before they can proliferate and potentially give rise to cancer. This self-destruct mechanism is a last resort to protect the organism from the threat of uncontrolled cell growth. By understanding and studying these processes, scientists hope to develop new strategies for cancer prevention and treatment. The development of cancer is a complex process that typically involves the accumulation of multiple genetic mutations over time. The two-hit hypothesis proposed by Alfred G. Knudsen in 1971 posits that two mutations or hits are required to inactivate both copies of a tumor suppressor gene, paving the way for cancer development. While inheriting a single mutated copy of a tumor suppressor gene increases the risk of cancer, it does not guarantee the disease. A second hit to the remaining normal copy is required to fully disable the gene's protective function. While mutations in oncogenes and tumor suppressor genes play a central role in cancer development, they do not tell the whole story. Epigenetic modifications, heritable changes in gene expression that occur without alterations to the underlying DNA sequence, add another layer of complexity to the cancer puzzle. These modifications, including DNA methylation and histone modifications, can silence tumor suppressor genes or activate oncogenes, contributing to cancer development. Unlike genetic mutations, which are permanent changes to the DNA sequence, epigenetic modifications are potentially reversible, offering promising avenues for therapeutic intervention. Cancer cells do not exist in isolation but rather reside within a complex and dynamic ecosystem known as the tumor microenvironment. This environment, composed of surrounding cells, blood vessels, immune cells, and signaling molecules, plays a critical role in tumor growth, progression, and response to therapy. Cancer cells can manipulate their surroundings, recruiting blood vessels to provide nutrients and oxygen, evading immune surveillance, and even promoting metastasis, the spread of cancer cells to distant sites. Understanding the intricate interplay between cancer cells and their microenvironment is crucial for developing effective therapies. Targeted therapies exploiting cancer's weaknesses. The discovery of oncogenes and tumor suppressor genes has revolutionized cancer treatment, paving the way for the development of targeted therapies. Unlike traditional chemotherapy, which kills rapidly dividing cells indiscriminately, targeted therapies exploit specific vulnerabilities in cancer cells, blocking the growth-promoting signals driven by oncogenes, or restoring the function of tumor suppressor genes. These therapies, often designed to target specific molecules or pathways involved in cancer development, offer the promise of greater effectiveness and fewer side effects compared to traditional approaches. Personalized medicine tailoring treatment to the individual. Cancer is a heterogeneous disease, meaning that tumors from different individuals, or even within the same individual, can harbor distinct genetic and molecular profiles. This inherent heterogeneity underscores the need for personalized medicine, an approach that tailors treatment to the unique characteristics of each patient's tumor. By analyzing the genetic makeup of a tumor, clinicians can identify specific mutations driving cancer growth and select therapies most likely to be effective. This targeted approach minimizes unnecessary treatments and maximizes the chances of success. The future of cancer treatment, hope on the horizon. The field of cancer biology is rapidly evolving, with new discoveries constantly reshaping our understanding of this complex disease. From immunotherapy, which harnesses the power of the immune system to fight cancer, to gene editing technologies like CRISPR-Cas9, which hold the potential to correct cancer-causing mutations, the future of cancer treatment is brimming with possibilities. The importance of continued research, unraveling the mysteries of cancer. Despite remarkable progress, cancer remains a formidable foe, demanding our unwavering attention and continued research efforts. Further investigation into the molecular mechanisms of cancer, the interplay between tumor cells and their microenvironment, and the development of novel therapeutic strategies is essential to alleviate the burden of this devastating disease. By unraveling the mysteries of cancer, one gene, one protein, one pathway at a time, we can pave the way for a future where cancer is no longer a death sentence but a manageable chronic condition, or even a preventable disease.